First of all, I would like to thank the organizers of this EPTRI open meeting for giving me the opportunity to present the data with our pediatric animal model. In this short presentation, I would like to highlight some of our findings with neonatal and juvenile Göttingen Minipigs in view of pediatric drug development. I am Steven van Cruchten, Principal Investigator within the Comparative Perinatal Development Group at the University of Antwerp. Dose precision in pediatric patients up to 2 years of age is challenging. In this respect, pediatric animal models can be very valuable, not only from a safety point of view when developing new drugs, but also to explore disease conditions in the pediatric population and as such potentially find new drug targets. In our research infrastructure, we are using the neonatal and juvenile Göttingen Minipig as an animal model for 3 different purposes. Neonatal and juvenile Göttingen Minipigs are used in first instance to better predict exposure of small molecule drugs in the pediatric population in order to avoid over or underdosing of patients. Therefore, we are gathering several physiological data in the Göttingen Minipig in order to develop a so-called physiology-based pharmacokinetic model. This can then be made available to several academic and industry groups for better dose setting in preclinical and clinical studies. Besides small molecule drugs, we are also investigating single-stranded oligonucleotides in the neonatal and juvenile Göttingen Minipig, and these are new modality drugs that are intended to inhibit the translation of disease proteins, for example in cancer. Typically, non-human primates are used as a non-rodent model of choice for safety assessment of these SSOs, but recently the adult Göttingen Minipig has been proven to be a suitable alternative, and in our group we are currently qualifying the juvenile Göttingen Minipig for pediatric safety testing of these SSOs. Finally, we are also investigating disease states in the neonatal Göttingen Minipig. In our facility, we are investigating perinatal asphyxia in this model. More specifically, we are inducing this condition in newborn piglets and assess the pharmacokinetics of several drugs with and without cooling therapy, as is done in the clinical situation. The aim is to assess whether cooling and/or the disease state affects the exposure to the compounds and whether dose setting needs to be adjusted under these clinical conditions. So, to achieve these objectives, we are using Göttingen Minipigs of several age groups. When you look at the table on the left side, you can see that we are using animals immediately after birth up to 20 days of age, and we use both sexes. The depicted age range covers the human pediatric population up to 2 years of age. In addition, we have also included some fetal stages to cover premature birth in men, and adult animals have also been included as reference value. So far, we have measured several parameters in these age groups, such as the pH and length of the gastrointestinal tract, organ weights, but we also assessed drug metabolism in this young population, and these data were used to feed into a PBPK model. In view of the single-stranded oligonucleotides, we assessed the ontogeny of endo and exonucleases, as the unmodified single-stranded oligonucleotides are prone to be hydrolyzed, this means metabolized, by the nucleases in the blood and organs. In the in vivo study, we also, of course, evaluated toxicity and included parameters such as clinical observations, body weight, clinical chemistry, etc. Finally, we set up a neonatal asphyxia model with and without cooling therapy, and in the following slides, I will take you through some of these results. As in the human pediatric population, we also noted in juvenile Göttingen Minipigs a fast decrease in gastric pH, in this case from postnatal day 1 up to postnatal day 28. This can have repercussions on the absorption of drugs depending on their physicochemical properties. We also noted high relative organ weights during the first week of life, and this was followed by a decrease up to weaning, which was postnatal day 28. There was a fast increase in absolute organ weights up to 3 or 4 months of age, and body weights increased linearly up to 5 months of age, which was expected. All these data can be fed into a PBPK software module, and this will help in better dose prediction. Of course, these morphometric data are not sufficient, and especially data on drug metabolism are also very valuable. These data will be further discussed in the next slide. In this slide, you can see 4 graphs representing hepatic metabolism of 4 different drugs by neonatal and juvenile Göttingen Minipigs, which are all depicted in the x-axis. In the y-axis, you can see on the left upper side the formation of acetaminophen, a metabolite of phenacetin by CYP1A2. On the upper right side, you can see the formation of 4-hydroxytolbutamide, formed by CYP2C9. And on the left bottom side, there was the formation of dextrofan, the CYP2D6 metabolite of dextromethorphan. And on the right bottom side, you have the formation of 1-hydroxymidazolam, the CYP3A4 metabolite of midazolam. For all drugs, we clearly see an ontogeny profile with the highest formation rate in adults, but for some of these compounds, we see a fast onset, and these are the ones on the left side, whereas for the drugs on the right side, we see a slower onset of metabolizing capacity. And again, these data need to be taken into account when we are exploring younger animals in our studies. We have also assessed phase 2 metabolism in our group, and these data have not been included in the slide, but you can find them back in the below publication. As I mentioned in the objectives, we are currently also qualifying the neonatal and juvenile Göttingen Minipig for safety testing of single-stranded oligonucleotides. In 2017, another group has shown that the Göttingen Minipig is a worthy alternative to non-human primates. And as these SSOs are also used, for example, in child cancer, we are currently investigating the younger age groups. For SSOs, no overt toxicity has been observed, neither in adult or younger age groups. The other data are still under further analysis, and so far we only can show you some data on the ontogeny of nucleases. So as an example, I have included this graph, and these are the results for 3 different exonucleases in the blood of different age groups of Göttingen Minipig. The values are relative to adult values. As you can see, there is a clear maturation profile for all the 3 exonucleases that are depicted in the x-axis, and this needs to be taken into account as the younger age groups appear to be less able to hydrolyze, this means metabolize, these SSOs, and this can lead to higher exposures for the same dose in these younger age groups. Further data can be found in the reference on the left side. Finally, we are using a neonatal Göttingen Minipig asphyxia model for perinatal asphyxia in human neonates. The piglet is a well-recognized translational model for this condition, as you can see in the paper on the left side. However, these authors use the conventional pig, and in pharmaceutical industry, the Göttingen Minipig is used. Therefore, we are setting up a model in the Göttingen Minipig. We are using a similar setup as our Danish colleagues, and the setup is shown in the picture on the right side. And we will assess the effect of cooling therapy on the pharmacokinetics of midazolam, phenobarbital, topiramate, and fentanyl, which are all 4 compounds that are used in a neonatal intensive care unit setting. These data will be fed into a PBPK model to better predict exposure and to have a better dose precision in this population. So, to conclude, so far we have already shown that drug metabolism of small molecule drugs and gastrointestinal parameters are similar in neonatal and juvenile Göttingen Minipigs compared to the human pediatric population. These data can be very useful for better dose prediction in this population. Second, the neonatal and juvenile Göttingen Minipig also appears to be a valuable model for new modalities such as single-stranded oligonucleotides. And third, the neonatal Göttingen Minipig also provides opportunities for assessment of covariates that cannot be addressed in a clinical setting, and we use perinatal asphyxia cooling therapy as an example. Finally, I would like to thank all our academic and industry partners and also our Flemish funding agency for their trust in our group and their support. And I want to thank you for your attention.